Welcome back. In this video, we're going to look a lot closer at some of the microscopic anatomy of the kidney. So before we get started, since you've already had this material in lab, I'd like you to look at this picture and look at the list of terms and make sure everything coordinates. And I color coordinated the words with the same colors in the drawing. So make sure you understand all those concepts before we get started. Okay, so let's get started, and we're going to learn about something called the renal corpuscle. And the renal corpuscle is actually two structures put together. It's the glomerulus and the surrounding Bowmans capsule. So how will you know if we're asking you renal corpuscle versus glomerulus or Bowmans capsule? Well, if the corpuscle is cut in half so you clearly can see the glomerulus and the capsule, if we're pointing at one or the other, then you know that's the structure. But on some of your other models, you have these ball-like structures which have not been cut in half. So like there's one here that was cut, and all these other, in this particular model, white balls, each of these would represent a renal corpuscle. So looking at the two components of the renal corpuscle, the outer Bowmans capsule, also sometimes known as the glomerular capsule, actually has two layers. What we are looking at as the outer capsule is actually the parietal layer of this, which is a simple squamous epithelium. There is an inner layer which covers the surface of the glomerular capsule, and I'll be talking about that a little bit later. I do want to point out that when you look at the corpuscle or the capsule, what you will notice at one end, this is where you find the blood vessels entering and exiting. So this end is called the vascular pole. In contrast, the other end is where the tubes are exiting. So this is called the tubular pole, or sometimes the urinary pole. So here's another picture showing the glomerulus cut in half, where you clearly can see Bowman's capsule with the vascular pole on your left and the tubular pole on your right. And inside the capsule, what we have now is the glomerulus. Now, the glomerulus is a capillary bed, but it's a capillary bed where no gas exchange occurs. So to reiterate, we're going to have an arterial carry arterial blood into this capillary bed, and we're going to have an arterial carry arterial blood out of this capillary bed. So between the capillary bed, the glomerulus, and Bowman's capsule, there is a space called Bowman's space. And the filtrate from the blood in the glomerulus enters this space. So looking at a light microscopic photograph, the upper pink one, you can see that it's actually quite a significant space in there. And the same thing is demonstrated with the scanning electron microscope picture below it. Looking closely at that glomerulus again, what we can see is it is covered by cells that don't look like your normal simple squamous epithelium. These cells are called podocytes. Okay, and so this is the visceral layer of Bowman's capsule we were talking about before. Looking at these podocytes, that prefix pod, P-O-D, like podiatrist, refers to feet. And it looks like these foot cells have like little toes or little digits that interdigitate with each other. So the ones from one cell will interdigitate with ones from another cell. And each of these little toes are cytoplasmic extensions known as pedicels. So between the interdigitated pedicels, there are little small spaces known as filtration slits. So whatever leaves or exits the glomerulus has to travel through these filtration slits in order to get into Bowman's space. So the picture on the bottom right basically has colorized some of the podocytes in the peachy color and some in the tan color. So you can see how they interdigitate with each other. Their pedicels interdigitate. Looking closely at the glomerular capsule, there's a cartoon drawing in the upper left, but there's a real scanning photomicrograph in the right side. And so take a moment and look at this and try to figure out which of the three types of capillaries you think this is. And if you have it narrowed down to a couple, but you're not quite sure, here is another scanning electromicrograph showing the beautiful podocytes on the outside, but it actually shows a red blood cell within the capillary, so you get an idea of sizes to help you figure out what type of capillary it is. Continuing on from Bowman's space, the filtrate next goes into the proximal convoluted tubule. Proximal because it's closest to the glomerulus, and convoluted because it's not a straight line. It's very wiggly, okay? It has, when you look at pictures of the proximal convoluted tubule, these are cross sections or oblique sections going through the tubules, and all the tubules have a very prominent basement membrane, which is this distinct purple line on the outside of each of these. You can see the lumen in the center of each tubule, and then between the basement membrane and the lumen, you then can see what the epithelium is and decide it for yourself what you think the epithelium is. Now, looking at the luminal apical surface of this epithelium, you can see that it's kind of like a bright pink, really fuzzy kind of thing, and that's because it has really tall microvilli, really tall microvilli. Guess what the microvilli are doing? Yeah, same thing microvilli do everywhere in your body, okay? In addition, these cells are chock full of mitochondria because there's some ATP that's going to be needed. What happens in the proximal convoluted tubule is the vast majority of the stuff that was filtered from the blood actually gets returned back to the blood while it's still in the proximal convoluted tubule. So most of your substances, think of your ions, all that kind of stuff, and a lot of your water, and your glucose does too, if you have a normal blood sugar. And the reason for that is glucose requires a carrier protein to get back into the blood. And so there's only enough carrier proteins for if you have normal glucose levels, but if you have a glucose level that's 3 times normal, that means you're gonna have glucose that's going to stay in your urine and have its own osmotic pull. After the proximal convoluted tubule, the filtrate next goes into the loop of Henle, which is sometimes known as the nephron loop. And here, even more water and sodium return to blood. And what happens here is really fascinating when you get to learn the physiology of it. I want you to realize from the proximal convoluted tubule, this loop of Henle is going to dip down into the renal pyramid in what's called the descending limb of the loop of Henle. Then it has a hairpin turn and comes up in the ascending limb of the loop of Henle to be in the cortex once again. So it's not called the descending loop and the ascending loop. It's the descending limb of the loop of Henle, or of the nephron loop. Once it has returned back to the renal cortex, you are now in the distal convoluted tubule. And let me go to the previous picture to show you something, just because it's a less complicated picture. If we follow from the glomerulus to the proximal convoluted tubule, down the descending limb of the loop of Henle, up the ascending limb of the loop of Henle, now we're in a convoluted, distal convoluted loop. I want you to notice that the distal convoluted loop always goes to the vascular pole of the renal corpuscle. All right, so now looking at cross sections, I hope from a glance, if you step back and try not to look at the P's and the D's, you can notice that, that some of these cross sections, the cells look decidedly pink, whereas other ones, the cells look decidedly purple by comparison. And the reason for that is in the distal convoluted tubule, although the cells have mitochondria, they don't have nearly the number of mitochondria as found in the proximal convoluted tubule. And you'll also notice that their cells seem to be packed closer together, and that's just because the cells are smaller in size. So I went ahead and put P's and D's in the lumens of these so you clearly can tell the difference. The proximal convoluted tubules are pinker, they have less cells, and they have that brush border from the really tall microvilli. In contrast, the distal convoluted tubule cells are purplier because they have less mitochondria. They don't have that brush border, and the cells are packed closer together. So at this point, this is the last chance for stuff to move from the blood into the distal convoluted tubule. And there are some significant things that move in this location, such as penicillin, which is too large to exit previously, now can exit the bloodstream. And in fact, 90% of the penicillin that's in your blood will exit in your urine, each pass through your kidneys. So I want to draw your attention to something called the juxtaglomerular apparatus, as in something is juxtaposed. It is next to the glomerulus. And so this is the area in the yellow circle down here. And so you can tell from the arrow that this is the afferent arteriole and this is the efferent arteriole. But there's two other ways to tell the two of them apart as well. One is more blood is coming into the glomerulus than leaving it because of the filtrates taking the fluid out. So the afferent arteriole usually has a wider diameter than the efferent arteriole. And the second one is the afferent arteriole has these specialized smooth muscle cells in its wall. And those smooth muscle cells are called granular cells. And they are in contact 24/7, chatting it up with some specialized cells that were found in the distal convoluted tubule. If you recall, I pointed out that the distal convoluted tubule always goes back to the vascular pole, and the reason for that is there are specialized cells, which you can see are a lot denser than the normal cells in the distal convoluted tubule. And those specialized cells, that area is called the macula densa of the distal convoluted tubule. And these are the cells that are talking to the granular cells of the afferent arteriole. And so this is the area known as the juxtaglomerular apparatus. And what they're talking about is not the football score of the day or how you did on your test or anything like that. What they're talking about, it has to do with blood flow. And if blood flow is down because blood volume is down, then the distal convoluted tubule says, okay, we won't pee out as much liquid. We will send some more back into the bloodstream to try to increase blood volume. If on the other hand, the granular cells are saying, man, blood pressure is sky high today, then the distal convoluted tubules would say, okay, we're going to pee out some extra volume to try to lower the blood pressure back down to normal. And so this is going to be super important process that we're going to be talking more about later on. So the juxtaglomerular apparatus is the site of a production of the hormone renin, and that's because renin has to do with this mechanism of blood pressure and blood volume regulation. Now, we also know from the kidney erythropoietin is produced. And until about 5 years ago, they thought it came from the juxtaglomerular apparatus. And now we know it doesn't come from there. We're not exactly sure what cells where do it, but we know it's not the juxtaglomerular apparatus. And we do know that it's in there someplace. So at this point, I have some photomicrographs that I want you to look at. And so when you look at that, I think you clearly can see some tubules going on. This is a glomerulus. This actually is an afferent arteriole leading into the glomerulus. And then do you see this tubule right here? See how it's darker than this tubule? And it's got a bigger lumen without the fuzziness. So this would be a proximal convoluted tubule. This would be a distal convoluted tubule. And you can really tell this is a distal convoluted tubule because you can clearly see the macula densa right next to the glomerulus. All right, so take a moment and look at that. Did you get all of it? Fantastic. Now pause your video and look at this one and see if you can figure and find out some proximal convoluted tubules, some distal convoluted tubules, the glomerulus, the macula densa. Yeah, just those 4 things. So how'd you do? Is this where you came up with? That's good. Now I got one more for you. Okay, by now you're a pro at it. And I'm sure all of you could find that area as the, what was it called? The juxtaglomerular apparatus. Fantastic. All right, continuing on after the distal convoluted tubule, now we have urine. I'm no longer calling it the filtrate because now we are in the collecting duct, which collects the filtrate from several nephrons. Actually, it's not quite urine yet because antidiuretic hormone works here in the collecting duct. And at night, you secrete more antidiuretic hormone. That's why you can sleep longer because you're not producing as much urine, and your urine in the morning is more concentrated. All right, once we are done with the effects of antidiuretic hormone, then we're going to call it urine, and the urine continues into the papillary duct, which is in the renal papilla, which then opens into a minor calyx, which may or may not go into a major calyx before it goes into the renal pelvis. So we are ready to excrete urine. So what is urine? We have this original filtrate. We sent tons of it back, and then we actually secreted a little bit more. That's what urine is. So it's what's left of the original filtrate plus what was secreted into what was left of the original filtrate. And this enters the renal pelvis to be excreted. So when we look at urine formation, in the most basic physiology, there's going to be 3 steps. So let me explain this diagram to you. This is a cortico-radiate artery. This is an afferent arteriole and an efferent arteriole going to a capillary bed. This is a stylized Bowmans capsule. So that this is the Bowmans capsule area in A, and then in B, this is proximal convoluted tubule going to loop of Henle, and then in C would be distal convoluted tubule. So basically, they just kind of straighten it out just so that it was a good diagram because it's the way I can put words on the other half. So the 3 steps of urine formation, the first one is glomerular filtration, which obviously occurs when the glomerulus filters fluid into Bowman's space. Pretty straightforward. The second step is called tubular reabsorption. Now, this kind of confuses students because when they look at this, they think, oh, stuff's going into the tubule, when in fact, that's not what it means. What it means is stuff that is in the tubule is being reabsorbed back into the bloodstream. So it's leaving the tubule, specifically proximal convoluted tubule and loop of Henle is where tubular reabsorption occurs. And then that is followed by tubular secretion, which means stuff can be secreted from the blood to enter the tubule. And this is what occurs in the distal convoluted tubule and to a minor extent in the collecting duct. So those are the 3 basic steps in 2 words each. So at this point in time, to see if you understand all this, draw a nephron and its associated blood vessels and see what you can label. In order to label the associated blood vessels, you're going to have to put that nephron someplace in a kidney. So you got to figure out what type of nephron you were drawing. So you see, the question is a little bit more complicated than it appears at first glance. Anyway, we are done with this section, and I will see you again as we continue our exploration of the urinary system. Thank you for all your hard work.